Welcome to another episode of the Dendroid Podcast with us, Saeed and Farno. This is a conversation with Dr. John Baenziger. He is a professor at the Department of Biochemistry, Microbiology, and Immunology in the Faculty of Medicine at the University of Ottawa.
His lab is broadly interested in understanding the role of ligand-gated ion channels in human health and diseases. These proteins located at synaptic junctions convert chemical signals into electrical response and are implicated in a wide range of conditions. They are also major targets for many pharmaceuticals.
Current research in the Baenziger lab focuses on the muscle-type nicotinic acetylcholine receptor and its role in diseases such as congenital myasthenic syndrome, myasthenia gravis, and muscular dystrophy to contribute to the development of new therapeutics. His team uses a variety of biophysical methods, including electrophysiology and cryo-electron microscopy. We hope you enjoy this episode. And now, here is John Baenziger. All right. Hello, Dr. Baenziger. Good morning.
Good morning.
Welcome to our podcast.
Welcome.
Glad to be here.
So we've been trying to coordinate this for a month, and you've been to different conferences. I think you once went to France in the beginning of the year.
Yes. I have a collaborator in France, and I do like to visit him as often as I can, and I had hoped to stay for a while, but that didn't work out.
Well, glad to have you here now. And
well, it's good to be here. I'm here most of the time.
Thank you so much. Let me ask you a little bit about your background. How has it prepared you, or how has it led you to come to Ottawa and teach a great course?
It's complicated. I think often when you talk to people about their careers, you sort of expect this linear path, and I'm driven to do certain things. I've often likened my path to being a ball in a pinball machine, and I get shot up and you bump around, and eventually you find your way. I did my undergrad at Queen's in biochemistry, and I think what I was looking for most out of that experience was just to be challenged and to be pushed to solve problems, to do things like that. When I was at Queen's, I happened to have some lectures by someone who was, I guess you'd call a biophysicist, which is, I guess, what you would call me to some extent. And he was someone who used solid-state NMR to study biological problems, and I was really intrigued by that, and that led me to do a PhD with him, and things just kind of fell into place. I think some people have read extensively enough to really be able to look and figure out the future.
It's hard.
When I was younger, I certainly didn't have that maturity, and then there's always a lot of other issues too that impact on one's life, such as your family and commitments to family, and so those things in part led me here back to University of Ottawa. Just my family's here, and so it was something that I always wanted to come back to, or Ottawa was always somewhere I wanted to come back to, and then. I grew up here. I wasn't born here, but I grew up here. So like a lot of people, I've heard people say that they, want to come back and contribute to the world that gave them the opportunities that they had. So there were a number of factors that led me back here. In hindsight, I can say that the University of Ottawa is probably hasn't been the best place for my particular research interest, because it's just not one of our biggest strengths. But it's also given me a lot of space to find my way. So it's been good.
Yeah, I wanted to ask, how is it different studying and working in a university like U Ottawa, and also the ones you did your
every institution has its strengths and weaknesses, and there are good things and bad things. But I don't think it's one can say, well, what's the difference between U Ottawa and say Harvard, where I was? Because when I was in Boston, I worked for a prof who worked on the protein I still study now. He was located at the Massachusetts General Hospital, and it was kind of a weird environment because at the time I was there, he had this he had a lab that had labs and a few offices in this sort of isolated area right in the anesthesia department. So I'd walk down the hallway and there are all these surgeons and people in their greens for going into the operating room, and then go down to this little corridor into the lab where his lab was. And I wasn't able to take in as much the essence of what it was like being at Harvard per se. I mean, you still get an essence of that, but it's, in hindsight, it's probably one thing I wish I had done more. A lot of that depends on what it is you do and the institution. So, for example, if I was to choose one place that I enjoyed being at the most in my career, it's been at the Institute of Structural Biology in Grenoble in France. And that's because, in part, I have colleagues there that are good friends and we enjoy working together. But also, that institute has a lot of structural biology, a lot of technology, a lot of seminars, people coming through there who are top people in the field. And so there's a sense of dynamism in that institute, and being exposed to all kinds of new and interesting things in my area. And so, someone here at U Ottawa who say works in neuromuscular research, and we have a very strong neuromuscular research core of people, they might say that Ottawa U is just really a great place for that and for those reasons, whereas other places may be stronger in other areas. So, I mean, it was great being at Harvard. I think one of the lasting memories that I have of being there is coming from Ottawa and being this sort of very shy and probably not the most confident young Canadian who worked at NRC. And then I went to Harvard and I had colleagues in that lab who had done their undergrad degrees at Oxford and Cambridge and all kinds of places like this. And I realized that the lab that I did my training in here it was affiliated with U Ottawa, but really at the National Research Council of Canada over on Sussex Drive. The people who were there were outstanding, and I realized that my training here in that lab with those people was really far superior to a lot of the colleagues I had in Boston who had been trained in Oxford, and that really changed my perspective on things in terms of my sense of Canadian science and so on. And so that was really one of the most important experiences that I had.
It's really about like the people you're working with and the environment you're in, rather than like the name of the institute.
I think so. I mean, look, the reality is if I'm a prof at Harvard, I can attract, just by the name itself, I can attract people
more opportunities.
Much easier because they recognize that name more than the University of Ottawa. I have a student, a PhD student a former PhD student of mine is at Berkeley right now, and he sends me emails once in a while about the people who've come through, the Nobel Prize winners who've come through to give seminars, the symposium they have, the technology they have, and that's specific to the field that I'm in, or at least partially in. And so for sure you get places like that where the reputation attracts the best people in the world, and you constantly have that environment, and that does impact on the science and the people who are there. So there are differences. Ottawa U has grown a lot since I came here, and I think we need to continue to do that, especially given the current political situation.
Yeah. No, definitely Ottawa is really growing compared to past years. The traffic is crazy, I'll have to say. But so let me ask you this. So you said people you worked with kind of prepared you later on, but I guess if you want to go to Harvard, you kind of have to stand out, and because a lot of people apply. And so what do you suggest a student who's studying in U Ottawa to kind of do?
Boy, that's a complicated question. If I look back at my own situation, I think the lab that I was in at NRC was a very good lab, and there were very good people there. And I kind of soaked that up to some extent and developed my own skills. And so I think that's really an important aspect. You have to develop your own skills and your own CV to put yourself in that position. How do you do that? I think one has to be very focused on what you're trying to achieve and very disciplined in how you approach things. So my most recent PhD student, who is very, very successful, if you look at the publications he has, his publication record, his knowledge, he was very disciplined. He came in every day. He came to work at 8 o'clock. He left at 5. I don't know what his work ethic was like in the evenings or outside of work. But he put in that time, and he did it every day and regularly, and he really worked with me to help move his projects forward and get the publication record that he ended up having. I think there are always opportunities that come along, and one has to. So if I'm a student, I want to be successful and have that kind of a career where I can go to one of these places, high profile places. I think being smart about it, being disciplined, taking advantage of opportunities. This student I had, for example, he was able to get an NSERC doctoral scholarship that had a travel component. So the last 6 months of his, or 8 months, I guess, of his PhD, maybe it ended up being close to a year, he was able to go to another lab to work. And so I started collaborating with these people in France to work on structures of the protein that I study. And the last year I was there on sabbatical for 6 months, and he went there as well to the same lab as me. So we were both there, and that was an opportunity for him to really expand his research profile and the types of things that he did. And he jumped on that opportunity and really ran with it. So I think being aware of what it takes, and you should have if you're wanting to be a scientist and go to some of these institutions, talk to your supervisors, be aware of the literature, read as much as you can, prepare yourself, and be focused on moving your projects forward. And if you are good at those things, and when you get opportunities, those opportunities can arise. And some of them are just flukes. Like when I did my postdoc at Harvard, I remember one of the people I worked with who was talking to me, and I was starting to mull over what to do for my postdoc, and he came across this ad at Harvard for a position, and he was saying, this sounds kind of interesting, and I looked at it. And I went to talk to my official supervisor, and he said, "Oh, that guy's visiting us. He's just down the hall." So I talked to him, and I ended up going and working for him. So these sometimes opportunities arise, but you have to put yourself in position to.
Right. You have to be prepared, and once the chance comes, you just do it.
For sure. Take control of your situation, be a glass half full person, and realize there are lots of opportunities out there. And I think one of your questions that you sent me was about working abroad, and that's one of the great things I think in science is that you have an opportunity to sometimes go and do a postdoc in another country, it's one of the things that I've enjoyed the most. I'm not a big person for just traveling, for vacation traveling, but I love going to places and working in those environments because you really get a sense of what those countries are like and the day-to-day things of living in those countries. And having that opportunity as a postdoc or a research associate or a PhD student to go to another country is always a great opportunity just to learn how people approach science and life differently, and so I think you need to be prepared, but embrace those opportunities. I wish when I had gone to Boston I had stayed a little longer and family reasons brought me back. I often thought afterwards that I was too worried about, getting a job and, instead of just sort of embracing the moment and living the moment and not worrying about that.
So now maybe we can start talking about the field specifically about structural biology and what are the challenges of, first of all, why should we
Okay, so you're asking me what I tried to teach you in my course, right?
Exactly, like I'm having a flashback of the first couple of lectures. And,
I mean, so, most people who go into research, biomedical research, are interested in human health ultimately. And as I, try to emphasize in the course that I taught, ultimately, as you start to ask general questions about human health, you have to dig deeper and deeper into cellular and molecular biology and how those things work, because that's at the level where often diseases occur. And as I've sort of tried to emphasize in the course that I taught, proteins are the machines in your body that make life happen, and they're also the machines in your body that often don't work efficiently or effectively when you have a genetic disease. And so ultimately understanding human biology comes down to understanding these machines and how they work together. And most, not all, but a lot of drugs are designed, a lot of therapeutic treatments are molecules that bind specifically to proteins and alter their behavior or alter their function. And so really from a biomedical point of view, if you really want to understand human biology, a large aspect of that is understanding the molecules themselves and trying to figure out what is happening with these molecules, if they have a mutation that leads to disease, and ultimately, is there some way that you can correct that? And one of the examples that I gave in the course was the disease sickle cell anemia. And Linus Pauling in the 1940s or 50s showed for the first time that disease was due to a chemical change, a mutation of one amino acid to another in that protein. And it was the first time that people had ever shown that a disease can result from a change in chemistry of one of these small complex macromolecules. And the interpretation he had was, if it's a change in chemistry that leads to this disease, maybe there's a chemical approach we can use to actually treat that disease.
Right. Okay.
So ultimately, that's what make proteins so important. And they're also, I think from my perspective, I like the fact that, when you solve a structure of the protein, there's a lot of complications to that, and a lot of it is there can be errors in how you model a structure, but if you do it properly, that structure is something that's there. It will be there forever, and it's a level of working where you can be very specific and definitive in your interpretations, as opposed to some areas where you're still doing very qualitative assays. Things are changing and trying to interpret those changes in the complexity of a cell's metabolism, and so on. So I like the fact that it's, there's a fairly definitive answer, and the interpretations are pretty.
Yeah, I find that very interesting about the course, because in other courses, we're studying this pathway, and we see the diagrams with the little icons of proteins interacting with each other. And I remember I'm looking at them what does it mean? What is actually happening? What is this circle?
Well, that's one of the hard parts of cell biology. And biology is you have all these proteins, and people are trying to understand on a cellular level how everything is happening, and trying to do it with that great complexity, and versus the single molecule thing. And putting the two together and understanding both is not always so easy. I find it much easier to understand a single molecule, and when it interacts with a protein, you can quantify that interaction and what concentration it occurs. And when you get to these interactome maps and things, there's a lot of complexity underneath that. But people are making progress in all those areas, and it's all towards understanding biology, cellular biology better.
So one of the proteins that you're currently your focus is the nicotine receptor. Can you talk
Right.
a little bit about
Okay.
that and why
So-
do we even care about
Okay,
nicotine
so
receptor?
it's the nicotinic acetylcholine receptor is the protein. And it's a protein that I think, as we discussed in my class, is a protein that has been central to our understanding of receptors and pharmacology, in that it was the first protein that people ever realized that the protein was a target for chemicals in your body. So a ligand binds to a protein, a drug binds to a protein. The protein I study, as I said, is the nicotinic acetylcholine receptor, but it's actually a family of proteins. So there are many different nicotinic receptors in the human body. I study muscle type receptors, and they have a specific structural composition, but they're also neuronal acetylcholine receptors as well. And they're part of an even bigger superfamily of proteins that respond to a lot of different neurotransmitter receptors. And these proteins are involved in a myriad of diseases, of conditions, things like epilepsy, a disease called myasthenia gravis, which is an autoimmune disease, a muscle disease called congenital myasthenic syndrome. And so ultimately, why I'm interested in that protein is because it is implicated in these diseases. It's a model for the entire family that helps us understand about this entire superfamily of proteins, which includes GABA receptors, glycine receptors, and glutamate receptor, or not glutamate, but GABA, glycine, and serotonin receptors. So understanding how these proteins work is very important to understanding a lot of those diseases. And I guess the disease that's most related to what I do at this point in time is a disease called congenital myasthenic syndrome, which is a disease that results from impaired synaptic communication at a neuromuscular junction. And some of the mutations that lead to this disease occur directly in the protein I study, and they alter its behavior and how it functions. And so ultimately, part of that is understanding how that behavior is altered, and perhaps how we can, modify that behavior to ultimately better treat that disease.
I see. So how do you distinguish, or I guess target different receptors in different part of the body? Does it have to do with the receptor structure? Or
well, that's often what people are trying to do. So when you have different receptors, they have subtly different structures, and the question is, can you exploit those subtle differences to get specificity? And that's ultimately one of the driving forces behind developing therapeutics. Is you want a therapeutic that will target the protein that you want to target, alter its behavior in a certain way, but only target that protein and not bind to other proteins, which lead to side effects.
I see.
And so there's a question of specificity and a question of modulating that structure to achieve a certain outcome. And so those are the two questions that are really underlie a lot of drug development. And there are many ways one can do that, but one of the ways you can do that is by trying to understand the structures of these proteins, and then use those structures as templates to try and design therapeutics that alter their behavior in a certain way.
One thing that I'm really interested in is how we build some sort of tolerance to those nicotines when they bind. How does that happen?
You mean
building tolerance and the receptors is not as sensitive to nicotine.
You mean when you're in terms of smoking? That's a complex question, and
there had a whole lecture on that.
Yeah, well, we talked about it, but we sort of just touched on it, and when you smoke, there's a line that was used in a paper about 15-20 years ago that I really liked by a guy named Henry Lester, who's at Caltech, I think, and he said that smoking addiction starts with the inhalation of nicotine, or I forget exactly the phrase, but you get the binding of nicotine to nicotinic receptors in the brain, and that leads to addiction, and over 4 million or whatever the number is, preventable deaths in globally. So nicotine binds to a specific neuronal nicotinic receptor, and it binds with very high affinity to those neuronal receptors. It doesn't bind with nearly as high affinity to the muscle type receptors. Otherwise, if it did, I think if it did, nicotine addiction would be solved because you'd probably die, because it would prevent your muscle type receptors from functioning, and that would prevent your body's ability to breathe and contract and relax the diaphragm. Okay.
And I'm remembering the experiment with the frog.
Yeah, and just as an example, there are nerve agents in the First World War when we had chemical warfare. There's a particular agent that affected the muscle type nicotinic receptor, and that's how it actually led to I think rather gruesome deaths. So nicotine binding to neuronal receptors does lead to addiction. I think it's a complex process, and it's complex because nicotine has certain functional effects on the receptor, but it also acts as a chaperone in that it enhances expression of these proteins in nerves. So studies have shown that the levels of these receptors increase, and there are different theories about how nicotine actually leads to addiction, and it's not my particular area of expertise, and some of them have to do with the fact that they inactivate these receptors. So when nicotine binds, it's a very weak agonist, so that means it activates the proteins, but prolonged exposure also leads to what we call a desensitized state. And it's thought that desensitized state helps, is one of the things that helps mitigate some of the features, psychological features that cause people to smoke. So if you smoke, there's cognitive enhancement, but there's also it's supposed to be helpful in treating anxiety and different things like that. And so when people smoke it, some of the ideas are that they inactivate these receptors, and that's helping to calm people down. But then they cause an upregulation of the number of receptors. So when the nicotine clears, all of a sudden you have more receptors there, which exacerbate the problems with anxiety. And so you smoke again to kind of bring things down. But it's a lot more complicated than that, and we'd have to get into a lot of physiology.
Yeah. It's really interesting because like with some drugs, you don't even build tolerance. Like for example, psychedelics like DMT or psilocybin, which is really cool. And
one of the things that people who study nicotinic receptors, especially in the neuronal ones, and in terms of how they affect cognition and different things like that, it's the one drug that we have, or I don't know, we have isn't the right word, but it is one drug that has neurological effects, but it doesn't affect your ability to function on a day-to-day basis. It doesn't impair your ability to drive, to work, and so on. And so it's the one drug that people have used for decades and decades and decades. And so we have a lot of just anecdotal information about how does nicotine affect different things, and there are correlations between people who smoke and benefits in Parkinson's disease, for example, and so on. So it's a unique thing because we can't do experiments on humans, but with nicotine we do have lots of data about how nicotine impacts human behavior and different human diseases. So it's kind of a cool field.
About the complexity of the field, do you yourself think about the murder mysteries when you're like dealing with these problems? One specific example you
I mean, so the example you're referring to is in class. I often, or I talk about our desire to try and understand how these proteins work, and it's kind of like a murder mystery, a detective who goes to a crime scene and tries to take all this information and put it together into a rationalization as to how that crime was committed, who committed the crime and why, and so on and so forth. And the reason I do that is because I often remember back when I was a student, I'd be in courses and I'd be learning about things, and I'd be sitting there going, "Why am I learning this? I don't get it." And that's probably a lot my fault. But I do try and convey to people or students in my classes that we're not learning all these details because I just love to go home and think about details. It's because those are the things that allow you to understand more complex problems. And so, in terms of sort of that detective analogy that I use in class, it's really about, taking these complex observations and putting them together in a meaningful way that we can develop understanding. And the reason I use often that analogy, which you will remember in class, is because, back 10, 15, 20 years ago, they had all these like Miami CSI shows, and they made detective work out to be, this really cool, exciting, sexy thing with, good-looking people and sunglasses and the whole bit, and we kind of really buy into that and think, oh, that's exciting, that's amazing, but in reality, the work is probably a lot of dogged, hard, very detailed work that requires a real attention to detail in how you approach things, and it's hard work, and so the idea that I was trying to convey is that we can look at things as being very kind of cool, but really there's a real hard work and detailed approach that's behind that all, and you have to kind of keep those two things in mind. It's not all it's the appearance is
not the whole picture.
Is not the whole picture, and you have to sometimes be prepared to really dig into things and understand at a real detailed level if you want to make the kinds of advancements that that you want to achieve the goals that you want to achieve.
But there's a saying like when you pay attention to details too much, it might prevent you to see the bigger picture. What do you say about that?
I'm not so. Look, I mean that's a matter of individuals and how. That's a matter of your talent, your vision, and so on. And yes, there are people who get lost in little details and seem happy to just play with details. And there are other people who are much more astute and see the bigger picture and really jump on those details and move things forward. And that's a matter, I think, of personal talent. But you can't ignore one or the other. I think it's. And you look at people who are extremely successful in science. They have a vision of where things are going, and they master those details and move on and use them to move things forward as best they can. And that takes a lot of discipline and a lot of commitment, and on a regular basis, and really knowing what you're doing and reading the literature and understanding. And having great ideas of where you want to go.
Yeah,
I don't think you can sort of ignore one or the other. It's just a matter of
just to balance it out
and being aware of what you're trying to go with what you do.
Knowing that is really important, because I remember sitting in class and as you were talking about different methods of how do we solve this structure, and you were saying that so this is how it works, but now at this point it's tedious. You have to put months, sometimes even and you have to be prepared for that. I was thinking this sounds interesting, but do I really want to go through this?
But at the end of the day, I think the question, and what I've come to realize in my own sort of journey in this career, is that if there is at the end of the day that really important goal that you want to achieve, at the end of the day you have to do the things that will get you there, and you can't shortcut that. If you do, it will eventually catch up, because you'll make mistakes. I'm thinking of solving a structure and how you have to go through and make sure every residue is modeled properly, and you can do that really quickly and kind of ignore things, but there'll probably be a lot of mistakes in your structure, and eventually people will start looking at your structures that you've deposited and go, this guy isn't very good. So there always is a balance, but it's a matter of how important is the goal that you're trying to achieve, and unfortunately that goal, or fortunately that goal, changes all the time. Like what you think was a huge amount of success one day, a year later, people may have blown way past that and gone to other areas. So.
Speaking of teaching, so which one is your favorite? I'm sure there's like pros and cons between teaching and research. Which one do you personally prefer?
I like both. I do more teaching, or I've typically done more teaching than a lot of people, a lot of my colleagues. I do like the teaching, but everything has its benefits and its not so great aspects. It's a lot of work, and it has changed over the course of my career. I'm at a stage now where I'm kind of going, okay, well, there are certain things I want to achieve with my research, and I don't have an unlimited time ahead of me to do that. So, it's becoming a bit more important to me, and I'm focusing more effort on that aspect. Whereas early on, I always just loved teaching, and I think say that. For example, in my course, I have a discussion group every week, which is, I get assigned a certain number of hours of teaching in that course, and then I do this discussion group, which is a lot of work and time every week. And I do that because I think it's important for students to not just be in lectures, but actually to start using the material. And
you read yourself, not just like a TA, like you show up again at 8:30 in the morning.
Friday morning at 8:30, it's wonderful. But and there's always a challenge. I've never been satisfied with my teaching. I'm always trying to improve the course, and there's an element of lecturing that really conveying concepts in a really clear and concise way that is always challenging, and one is always trying to achieve better. And I just as an aside, I think at times the students who want to take a 10-hour course or a 2-hour lecture on how to communicate properly, and I'm thinking I've been doing this for a long time. I'm still trying to improve how I communicate and still have to spend time on that. It's an ongoing process. So I like the teaching aspects. I think right now my research is a little bit more important just because of the timeline, but at the same, and part of that is probably because the courses that I teach, I've been doing it for a long time, and I think I have them set up in a pretty good way. But when I start teaching, I'm always glad to be in the class, and I always, when I start to prepare for a new course, go, oh yeah, I got to improve this and that. And so there's certain challenges. It's just a matter of juggling both.
Right.
And you could say that, like when you're a student and sitting in different classes, you can say the difference between how a lecture is structured. Because like sitting in your class, it is really complex, like material. But the way that I didn't take any notes, and like it took me time to understand that no, I really don't need to. I should just listen. And the way that the slides are set up, and like you're explaining how it's going, you could see the time and effort that has been put
and some of it's been stupid. I mean, there's one lecture where I have just a visual of how people used to do sequencing. And I just have this bar with a sequence and a Pac-Man that comes along and bites off a residue and there's supposed to be a crunch there, and it spits it out and tells what the residue is, and just to illustrate conceptually how that is worked, even though it's done chemically. And I can remember, one day it was over the Christmas holidays, I was just at a coffee shop and I was working on that, and I was thinking, how many hours did I spend on that? And I was sort of scratching my head, what am I doing here? It's the holidays and it's Christmas time, and I'm sitting in a coffee shop trying to get this little crunch sound to work on my little Pac-Man thing, and so sometimes I can get lost in that. But I think at the end of the day, I'm just someone who likes to be busy and engaged in terms of my mind, and so the teaching can do that as well as the research can as well. And so, there's a real fun aspect to doing that. And, some classes I find the students, there's variability from year to year, but particularly your class I found really a great class, and the students were really good and engaged. And and that. But you know, I'm also conscious. I've had one student who was in that class who works in my lab, and he says that when students look for rotation projects, they put a red across my name because they're worried that I'm going to expect too much of them.
Well, no, but that's the thing, because like you were completely honest about how the field is, and I was actually considering it, but I didn't know that. Well, it's like the same level of effort, the same level of like tedious work, the same level of like details is in every other field.
Yeah,
not
it's
even in
in
just
everything.
science. Like,
Yeah.
like I volunteered in some like in a community theater. I'm going there. I'm like, well, this is just like volunteering. But if you want to do that, you have to put like 3 months of commitment. You have to like practice every single day, and it's the same line.
Yeah.
It's the same thing.
I
It is
remember a colleague of mine who I visited in Switzerland, very well-known guy. He had 2 sons, and he told me that his sons told him that they didn't want to go into science because they saw how hard he worked, and they didn't want to do that. So they went into another field and found out that they were working even harder and getting paid a fraction of the salary that he was earning. So, you know, I think if you want to be good, you have to, you're going to have to work at whatever you do. But I think what is sometimes lost in that is it is a privilege in life to have a job and a career where you are engaged and are excited about doing what you do, to the point that, you know, if you work on a weekend a little bit on it or whatever, if you work on the weekend a lot, you actually are engaged in that. And to be engaged in your work for an entire career is pretty special. And so in some ways, you know, we talk about work-life balance, but I think there's something to be said or realized that if you're in a job that you're engaged in and you want to work more than, you know, sort of your 7.5 hours a day because you really like it, that's a pretty special thing to do. And you'll see that. I mean, actors will talk about that, and they're still acting in their 80s, and it's what they love to do.
Yeah, because actually that has been one of my concerns, and I think about it a lot, to how to balance life and work. But at some point you need to kind of disturb that balance for a while to be able to reach that balance in general and as a whole. So sometimes you got to work harder. Maybe you don't get enough sleep when you have to.
Yeah,
I guess
mean, those
prepare
are
for your exam.
tough questions,
That's
and they're
it.
very personal
Yeah.
questions. But I think being in a career where you're engaged is pretty special, and and that's one of the things. Being a professor, you know, I'm always conscious. As much as, you know, I might work very hard. I arrive at work at 7:15 every day, pretty much, and I'm here till usually around 5. So, you know, I do put in long hours, but I guess those are choices, and you know, you'll find kind of that balance. But there also is a time in your life when you're young, I think, you have to master what you're doing and become good at it. And you can't just say, well, no, I'm only going to do a work-life balance thing if that doesn't allow me to be good at what I'm doing. Because it is a competitive world, and it's becoming more and more a competitive world. And so one has to devote the time you need to kind of to get yourself in a position where you're a success. But, you know, being a professor and being able to decide what I do most days when I come to work and is, you know, that's a real privilege.
And it's interesting because like you see people like in the middle of their careers or who are quote unquote successful, and you ask them, well, looking back, do you have any regrets? And they think, and then, well, maybe I should have like taken more time with my family or like just live. But it's like, well, you are in the position that you are because of the work you've
You
done.
know,
Yeah.
but those,
It's like-
but that's life. I mean, you can always look back and say, I should have spent more time.
Yeah.
Do I wish I'd spent more? Well, you know, I could look at you can look back at any life, no matter what you did, and say, I wish I'd done more of this or done more of that. For example, I had kids when I was in my 20s, and that was hard to do a postdoc and to deal with all that. And so my kids were young when I was still kind of trying to get established, and financially, that wasn't so great. I mean, I remember we used to go camping in our little car. We'd have a canoe on top, bikes hanging off the back. I'd have to, kids were sitting on blankets and pillows in the back seat because our car was so small. And I compare that to people who had kids later on in life, where they were much better established financially and they could do a lot more stuff with them. But I also look back at those days, and they were kind of fun too.
Have your kids ever considered like doing what you're doing now, or?
Not really. My daughter had a science kind of bent to her, but her mom also worked in the Royal Ottawa Hospital, so she had kind of a little bit of a mix of science and mental health issues, and so she became a psychiatrist.
Oh, nice.
My son, he went into more programming, financial kind of stuff. So none of them really followed my footsteps. But my dad was a scientist, but he was a plant scientist back in the day, where they'd do some breeding and then sit and watch their plants grow. I'd probably be disrespectful to my father. I hope he's not looking down at me. But it wasn't the same as it is now. And but I like that challenge. I don't know where that came from. I think, as a kid, I was a competitive swimmer, and that was swimming 3.5, 4 hours a day, in the morning, 6 to 7:30 a.m. and then after school, 4 to 6, sometimes 6:30, depending on the time of year, and that was 6 days a week. So I've always liked, and maybe it stems from that to some extent, just being engaged and having things to do and being forced to be disciplined in my approach. I even remember in high school, I had to really, with all that time devoted to swimming, be very kind of organized in how I approached my schoolwork. And I kind of liked that to some extent. And I had to make up for the fact that I was always so exhausted and tired that I'd be sleeping in class all the time. But anyway.
But you have to sleep sometime.
Yeah, absolutely.
No, swimming is hard, and it's one of the hardest sports. Like, usually people don't realize it, but it's like definitely the hardest. Like,
the difference
with
one of the things with swimming is it's very technical. Like I do a lot of running, but anyone, you can go out and run, and your body has a certain way of running, and everyone does. But swimming is not that way, and it certainly makes me laugh, because there were times as an adult that I went back and did competitive sort of swimming, and you could see the people who trained when they were young, because they were in the pool 4 hours a day and learning the mechanics of it, and even if they don't train very hard, they're so much faster than people who at a later stage in life are trying to learn how to swim, and you can tell the difference.
How did you learn to swim? Because my experience, my dad just threw me in the pool, and it was like, you got to swim.
That's the beginning for everyone.
Yeah, I mean,
Or is it like that?
we had, I camped. We were always near the water when I was a kid, just running and jumping and playing in the water, and then somehow I started. I liked swimming. I started swimming. In Nepean, there are some outdoor swimming pools, and they used to have little kind of swim teams in the summer. They were very, kind of low key, but that led from one thing to another, and that, you tend to gravitate towards things you're good at, and for whatever reason, I was a pretty good swimmer and pretty fast, and kind of moved through that process very quickly. But then, getting back, then the hard work starting is 4 hours a day and constantly and all that stuff.
Do you still do a lot of swimming and running and camping?
Yeah. No, I'm very active. I'm trying to be more and more. I mean, I run more than I swim now, but that's it's getting harder as I get older to balance those things. 2 years ago I did an ultra marathon in France where I think they're starting to have them here. They're in France. They're kind of French people are a bit nuts. Like they have these races, and they're not just 20 kilometers or 40 or 80 or 120 kilometers, but they do it running up and down a mountain, right? And running up and down a mountain is very different than just running on a flat surface. So I did one of those, and so I do that. I cross-country ski a fair bit. I run. I do lots of activities. I like to do. I do a whitewater canoe trip every year. I mean, I'm always tend to be like things mostly around water. So whitewater canoeing. There was a while I used to surf a bit too, and love to do that more, but just no time these days.
Yeah. So back to research again. Recent Nobel Prize was about figuring out the structure of the proteins and the
with AlphaFold and protein design.
Yeah. So where we are now, and what are some sort of future plans or goals we're trying to achieve?
Yeah. So I think those prizes were awarded for fairly recent work because they have really changed a lot of what I do and what people will do moving forward. So with AlphaFold, you can take. People have tried to predict how protein structures for a long time, and the typical approach of doing that is more by trying to fold proteins and energetically have them adopt their lowest energy conformations. Whereas AlphaFold uses databases and artificial intelligence to try and predict structures, and it actually works very, very well. So if you predict a structure of the protein I study, it's actually very similar to the structures that we have solved. And that software has changed a lot of how you solve structures. You still have to solve structures because you can predict certain things, but you don't know how accurate those predictions are without a real structure. So it helps you in terms of actually solving structures because you can predict the structure ahead of time and then use that to help fit your maps and things. But I think the more important thing is the protein, the combination of that with protein design. Because you can now take a protein and try and design, for example, binders to that protein that will bind and affect its function in a way that is of therapeutic use. And you can do that without any knowledge, detailed years of structure and function. And there was a paper published a month or two, maybe it's a bioarchives paper, but no, I think it was published in Nature, where they use these approaches, for example, to develop anti-venom agents. So snake venom has a lot of things in it, but one of the things that it has in it is these toxins that are called, they're neurotoxins, which bind to muscle-type nicotinic receptors and some other nicotinic receptors as well. And people have been working on trying to develop antivenoms for a long time. What they did with this new technology is they said, okay, we now have structures, and some of them are my structures, or structures that I was involved in, of the muscle-type receptor, and we've solved structures with some of these toxins actually bound to them. So we know how they bind to the protein. And what they said is, okay, we'll take these toxins and we'll just use artificial intelligence to develop other proteins that bind to these toxins. And the idea is if they bind in a certain way, then they will prevent the toxins from binding to the target proteins and then can be used as antivenoms. And so they did this whole protein design approach. They developed a bunch of potential antivenoms and screened, in silico on a computer, and came up with a few antivenom agents that targeted the main types of toxins. And in one paper, they did this all and actually showed in mouse studies that these proteins they designed de novo using these artificial intelligence means were able to actually act as antivenoms much better than anything anyone has developed. And I look at those papers and I think of all the work that people have been trying to do to understand a lot of the basics, and some of that is critical for these artificial intelligence methods. So the protein itself, those structures are important. The toxin structures are important because they allow you then to design binders to these things. But it seems, in that short period of time, they went from nothing to having these much, much, much more effective antivenom agents. Now, I don't know in clinical situations how will they work. Will there be other problems? It's possible. But I look at that field, and I even had one collaborator who I was giving protein samples to who was trying to develop antivenoms, and he's been working on this for a long time. And I'm just thinking, like, this guy must be sitting there going, with his mouth jaw dropped, because all of a sudden, these new protein design methods have just wiped out his entire research program because of this advancement in technology. And this is one of the things we talk about in my proteins course, is technology is responsible for so many advances, and it's driven by the biology. And there's so many Nobel Prizes that have been given to people who worked on developing methods for solving structures, and now for protein design and predicting protein structures. As we get more and more structures, and that allows us to be much more predictive in terms of our approaches. And so it'll be interesting over the next 10, 20 years how that changes, how people fundamentally do research, and how it changes how people design therapeutics. And I think most people think it has already changed dramatically how that happens. So, and I get back to my teaching, I realize next year I'm going to have to nix one of my lectures and have a lecture at the end about AlphaFold and protein predictions, how it works and how people can use that. Even my collaborator in France, he is so the protein I study is called, as you mentioned, the Nicotinic Acetylcholine Receptor. It's also found in non-vertebrates, so it's found in bees, for example. And one of the big problems we have right now is that a lot of the pesticides that are used for crops are agents that bind to nicotinic receptors in some of these insects. And I'm not sure what the exact mechanism is, but it ends up killing these insects. And so you hear about things called neonicotinoids, which are agents that bind to nicotinic receptors in bees. And so one of my collaborators in France is now trying to develop for different types of insects, looking at the structures of the types of receptors that I study on, and then use these artificial intelligence methods to design binders that inhibit them, but that are much more specific. And the nice thing about these protein therapeutics is, unlike some of the chemicals which will stick around in the environment for a long time, these proteins are just proteins. So once they're exposed to the environment, they're just degraded and become completely innocuous. So. Right. So it is changing the world, and I think it's changing. It'll change very much biomedical research.
So would there a chance to kind of work towards desynthesization and kind of preventing that by introducing the binding sites or some sort of thing?
Sorry, say that again.
How we earlier talked about building tolerance and desensitization of those receptors.
Desensitization, yeah.
Would there be a possibility to maybe smoke cigars and not get addicted to them?
Well, I don't know. It's possible that there could be ways to treat that, for sure. I think people will be able to do that. I think, if you smoke cigars, there's a lot of other health drawbacks to smoking that...
What about
so I'm not sure you want to find some way of making them not addictive, and so people can smoke, given all the other health consequences.
But what about how people like Andrew Huberman, one of our most podcast influencer, he talks about taking nicotine as a supplement, just like pills, or nicotine gums.
Oh boy.
Is that like some sort of beneficial, or do you think
I don't know. I think everyone will have an opinion on that. At my stage of life, I certainly am becoming more aware and of things that may combat aging and keep you young and healthy and keep me allowing to run my ultra marathons if that's possible. But on the other hand, I tend to be someone who is very much a purist in that I try and take as little as I can. And when I go to doctor's offices now, they're asking, "Do you take this?" And I just no. And I think there are always consequences to taking drugs. Now the benefits can outweigh the consequences, right? But so I don't know. I think there are people online. I'm sure you can find them about who are. That famous guy in the States who's trying to live till he's 200 and so on. And what are the, and you look at his fridge and it's like a medicine cabinet and all these supplements. I think, those are tough questions. I don't have the answer to them. And I think some of them are personal, but
it's like one difference between the front line of research that, well, you have an idea, you like write a paper with it, it sounds promising, between and on the other side something that has been established. It's like, sure, that's interesting, but that's like very specific, and you have no idea about other aspects of it.
The reality is, it comes back to these structures and specificity. If you design if you have a small chemical that you ingest at high quantities, it may target a specific receptor and do so with high affinity. But if your levels of that drug get high enough, then they can also target other things and bind to other things with side effects. And ultimately, what are the consequences of those side effects? Those are some of the tough problems with therapeutics and and you hear stories about that in the news. I think there have been recent ones about drugs. Was it for Parkinson and the side effects that they have, and so I tend to think living healthy and eating fresh vegetables and
that still works.
That's the best, the thing that makes the most sense to me. But it's certainly something I'm interested in, especially as I get older, and you want to stay as young and active as possible.
Well, speaking of tough questions, let me ask you this: what is the meaning of life, and what is
great that you were going to ask me that.
What gets you going these days?
What is the meaning of life? I don't know. I'm not sure what that even question means. What is the meaning of life?
Like, what gets you going? And
what gets me going? I think for me, it comes back to this issue of, sure, it takes dedication for a career, but if it's engaging and that everyone is different. My daughter, she's a psychiatrist. She works actually very hard, but she would prefer not to work hard, and she likes to travel and she likes to do all kinds of things, and that's what really stimulates her and drives her. I think for me, I need to be engaged in things and projects. So I love the fact that my research, for whatever, some of it is fortuitous. The technology has caught up to me and allowed me to do things now that I wouldn't have imagined that I could have done 10 years ago. And it's still engaging and doing new and exciting things, and things that are new, and doing things that no one else has ever done. I mean, that's pretty exciting when you have a structure and no one else has seen that structure. And one of our biggest structures I've talked a bit about briefly in class. It's been published online now, but we're still waiting to hear from the journal that it's been submitted to. And And so that's exciting. There's always downsides. It's hard work, and it's stressful to keep all these things going and moving them forward. But doing new and exciting things is important. I don't know what I'll do as I approach, kind of my retirement. Whether I don't think I want to go off and hang out in Florida. I don't know. So I'll probably look for other challenges. And I think I think the important thing is to always look forward and trying to find new challenges and new things that engage your mind and that keep you busy. I think that's really important. And the other thing that I would say is, always try from a young age to live a healthy life and maintain your health. I know a lot of people my age friends who are retired who have a lot of health problems, and that's overwhelming their daily routines and thoughts. And I even had one person tell me, you should be very careful about getting all these things done now, because you never know when you're not going to be physically able to do these things. And this was a few years ago, and I said thank you, and then went off and ran my ultra marathon in France, and didn't worry about it. So I think those are two things, not necessarily the meaning of life. That's a bit of a more philosophical question, but the two things that I think can help you live a good life are maintaining your health and enjoying that aspect. So when I'm running and doing it regularly, and I can get any runner who's listening to this will know, you get to a point where you're almost like flying, the way you barely touch the ground, and there's a wonderful feeling. So to keep that physical aspect and health aspect is important, and then to be engaged. And then there's always the family aspects too. But I think, and it depends for everyone, but I know for myself I need to have things that force me to be disciplined and make use of my time that way. If I have too much time and I'm just a blob on the couch, that's not good for me.
What career would you choose if you were not a scientist?
I have no idea. I saw you mentioned that question.
Marathon runner, I guess.
No, I like doing those things, and I'm at my age. I can do pretty well, because not many people my age are doing the things I do. In fact, I went to see the doctor about a sore knee, and he made a comment about, well, if you're going to be doing stupid, crazy things like this at your age, and I said, what are you talking about? But I wasn't. I wasn't good, but I wasn't at that level, and I'm not. I didn't set out to become a scientist. I kind of ended up doing it because it's where I found challenges and things that I could get excited about and do, and I think that really led me to that. And so I don't know what I do. I would hope I would find something that's challenging and can, get me excited about doing stuff. But a lot of other careers don't give you that creativity, and that's one of the things that I don't think people generally think of scientists as being very creative in terms of what they do, but that novelty and creativity is, I think, what actually ultimately drives a lot of people who do science, and you can see that when you read certain papers and you look at what these people did, and it comes out of nowhere, and you go, wow, that guy's or that woman or that man or whatever is just brilliant, and what a creative way of thinking about doing things, and how did they come up with some of these ideas? And it's built on probably a lot of other people's work at the same time, but I would hope it'd be something where there's some element of that, because I think that's certainly what keeps me coming into work early in the morning.
Look, it was really inspiring to be in your class at every lecture. In the first class, I entered the classroom and you were talking about the importance of having an independent mind and think of yourself And I was like, well, this class is going to be important whether or not I'm going to stay like in this field or not. Like I'm going to remember some stuff.
It's
And
one of
this
the most
conversation
important
as well. Yeah.
Okay, well, good. It's been a pleasure being here.
Thanks for coming.
Thank you very
Okay,
much.
thank you.
Thank you.
Thank you for listening to this conversation with Dr. John Baenziger. In this episode, we discuss structural biology, muscle-type nicotinic acetylcholine receptor, specifically solving protein structures and life outside of the lab.
This is the Dendroid Podcast. To support it, please share this episode with your friends and anyone else who might be interested. And now, let us leave you with a quote by Linus Pauling that Dr. B told us the very first day in the class. When an old and distinguished person speaks to you, listen to him carefully and with respect, but do not believe him. Never put your trust in anything but your own intellect. Your older, no matter whether he has gray hair or has lost his hair, no matter whether he is a Nobel laureate, may be wrong. So you must always be skeptical. Always think for yourself. We hope you enjoyed this episode and see you next time.